Hello, everyone. We're pleased to have you join us. I'm Lauren Costi, Director of Business Development here at CDD, and as your host today, I'd like to welcome you all to our Collaborative Drug Discovery Quarterly Product Update webinar. So before we dive into the exciting updates we have for you, I'd like to quickly go over a few housekeeping items. For any product-specific questions, please use the Q&A tab in the toolbar. This will help us address your inquiries efficiently during our dedicated Q&A session, as well as answer any quick ones as we go. For all other questions, comments, or general discussion, please use the regular chat box tab in the toolbar. We encourage your active participation throughout the webinar. If you would like to request a one-on-one customized demo for any of these features, we'll have a poll to easily request one before the Q&A segment. So now let me give you a brief overview of what we'll be covering today. We have 4 key modules to discuss, each representing significant enhancements to our platform, starting with Curves. We'll explore new functionalities that will streamline your data analysis processing and help express complex trends in a concise manner. Jumping into AI, you'll learn about our artificial intelligence integrations designed to accelerate your research and decision-making. Next, we'll showcase updates to our inventory management system, improving searchability, traceability, and accountability. Last but not least, we'll walk you through the enhancements made to the electronic lab notebook, including templates and our new annotation tool. Each of these modules have been developed with your needs in mind, aiming to further optimize your efficiency as scientists. We're excited to share these updates with you. Your feedback is invaluable to us, so we encourage you to engage actively throughout the presentation. Thank you once again for joining us today, and let's begin our product update journey.
Hi, everyone. I'm Mel Berg, Account Executive here at Collaborative Drug Discovery, and I'm delighted to introduce you to our new Curves module today, a tool that is designed to make analyzing and visualizing your complex data easier. Curves will simplify your data processing and presents complicated trends in a clear visual way. So whether you're a biologist or a chemist, Curves will enhance your research. Let me highlight 4 points for you. Integration. You will save time and reduce errors because Curves provides an automated workflow that takes raw data and transforms it into plots. Presentation. Sharing, exploring, and exporting your visualizations will be much easier as Curves gives you complete control over the display options and a new color coding feature to enhance visual clarity. Comparison. The module allows you to search across assays to evaluate trends and overlay top hits, which is essential for identifying promising compounds. And collaboration, because you can securely share your visualizations within a project-based environment. And the new plot comments feature enables direct communication about specific data points. So let's quickly look at some key features of the Curves module. We have Curve Overlay, where you can compare multiple results on a single plot with enhanced color coding. Summary Curve, which is a single toggle, allowing you to summarize results over time for quick decision-making. Curve Fits is a module that offers various curve options for data analysis, including newly optimized bell-shaped fits for complex biological responses. And calculations, so you can create plots using raw, normalized, and calculated values. So whether you're plotting potency, selectivity, ADME, toxicity, PK, or PD data, Curves will help you tell a clear and compelling visual data story. Our latest updates in color coding, bell-shaped curve fits, and plot comments will further improve your ability to gain insights from your complex data. Now, let me hand it over to Ralphie for a live demonstration of these features, including the latest updates. Ralphie, take it away.
Hi, everyone. My name is Ralphie Fiorenza, and I'm a customer engagement scientist here at CDD. Today, I will be stepping into the role of a biologist running a ternary complex assay against a panel of selected PROTACs, and this will be to showcase CDD's new Curves module. As you can see here on the screen, I've been able to fit a bell-shaped curve and then calculate an AC 90 and 99 based on my ternary complex assay data against these selected PROTACs. And how did I get here? In order to utilize the Curves module, it has to be turned on, and then from there, your Vault administrator would go to Settings, Vault, and Plots, from which they could select from a number of curve fit algorithms available at the time, such as the Hill and Bell equation, and they could even add in lists and pick lists of curve comments to help you all through your QCing process. So once you have the actual curve fit algorithm enabled, you would then go in and create a protocol just like any other protocol. So here I have my ternary complex assay protocol, and then I have my readout definitions corresponding to the data coming out of my particular assay. In this case, I have the concentration and luminescence being captured. I've then asked CDD Vault to please normalize my luminescence to controls on my plate so I don't have to do any sort of pre-processing of data. And then I've captured some additional readout definitions of interest to me, like the cell line and time point of the experiment. Once I have all of those readout definitions captured, I would create a new readout definition with the data type of plot. And this will allow me and ask me to choose my X and Y for my plot, in this case, concentration and fraction of maximum. In addition, I can actually set a default y-axis range. So for here, I've done -0.2 to 1.2 for this particular assay. I've chosen my curve fit algorithm as the bell curve, and depending on the curve fit algorithm that you choose, you will get different options here in terms of your constraints and data calculations. So since I've selected a bell curve equation, under my data calculations, you'll see that I can actually set an intercept on the left side of the curve or the right side of the curve. For this particular assay, I'm interested in my near max response, so I'm going with an AC 90 and 99. Once I have my protocol set up with all of my readout definitions and normalization, I would then go to my Import Data tab and choose my data file. So in this particular example, I'm going to be uploading a raw data file that has not been touched or processed. So it's just right off my plate reader with my concentration, response, plate layouts, etc. So I can choose to just upload this exact file to CDD. And what will happen is we can actually utilize the built-in file parser. So here I have a pre-saved parsing template for my ternary complex data. I simply click Upload File. CDD Vault will take us to this page where we've organized the data into that tabular format, and then automatically take us to the mapping step for our protocol readout definitions. I could choose to individually click through and map my plate and well, cell line, etc. information, but what I've done in this particular case is I've saved a mapping template to further automate this process for myself. So I would collect bell curve mapping, create a new run, and then apply the mapping template, and then all of my mapping has been done for me here in green. All that's left for me to do is click Process File, accept the import, and then we've gone from raw data parsed by CDD and actually analyzed and fit to a curve in a matter of seconds. So what do I do now? I'm looking through all of my data here, and I've decided I actually want to export a graph with a few compounds on it of interest to put into a presentation or send off to a key stakeholder. So in order to do that, I would just have to click on a particular curve of interest. That will open the Curve Mini app, from which you can individually interact with the curve. So editing outliers, so flagging your outliers, overriding the curve fit parameters, and even adding those QC comments that I told you about earlier. But what I'm really interested in is actually creating an overlay of two compounds. So I would click this little plus button up here at the top. A window opens up, and I can add multiple molecules as well as multiple protocols to a single plot. So in this case, I'm just going to type in the name of a molecule that I'm interested in, and I'll do 2 molecules, 1 protocol, launch plot, and we'll open up a new tab, and you'll see in the carousel here, I have my 2 compounds. I can change the color, shape, and size of my individual curve data points to make it more distinct. And then when preparing my export, I would click this settings button. I can hide those grid lines as well as the intercepts for the AC 90 and 99, and even add a chart title. And then if I go under the statistics tab, If I had replicates in this particular protocol, I can choose to show my whiskers, and I can add an arbitrary line to call attention to a specific point on the plot. But I'm very happy with this particular setup. And now for export, I can do PNG or PDF. And once I select one of those, I get additional configuration options. So here I can customize my X and Y axis. I can choose to export the structure in the legend, and then really just pick and choose any of the information that's relevant to your export. Click Download PNG, and it'll go to your Downloads folder. And just like that, we've gone from a raw data file that's been automatically parsed, processed, curve fit, and overlaid by CDD Vault in a matter of minutes, really streamlining my workflow here. And that's all for curves right now. If you would like a personalized demo for your team, science, and workflows, please make sure you vote in the poll at the end of this webinar. And next, we'd like to introduce you to CDD's AI.
Hello, everyone. I'm Eric Gifford, and today I'm excited to introduce the Collaborative Drug Discovery AI module. The AI module can help you increase the speed, accuracy, and efficiency of your drug discovery and development programs. Our AI module is seamlessly integrated into your existing CDD vaults. This integration allows you to harness advanced AI capabilities without ever having to leave your safe and secure CDD Vault environment. Let me highlight just 3 of the benefits that make it so powerful. 1, increased efficiency. Our AI algorithms enable researchers to quickly identify related, synthetically feasible chemical structures in a collection of over 30 million publicly available compounds. 2, enhanced precision. The results of these searches allow scientists to develop testable hypotheses and more accurate bioactivity and molecular properties predictions by focusing on the most relevant sets of compounds and related data. 3, data-driven insights. Our AI uncovers patterns in large data sets and presents the results in a way that enables scientists to suggest testable hypotheses. In summary, the AI module's key capabilities include fast AI-powered searching for exploring compounds within Chembl, Enamine, and CDD Public, as well as patents within SureChembl. Intuitive results presentation, including clustering and scaffold identification of the related compounds. Bioisosteric replacement suggestions to enhance potency and improve your properties of your lead compounds. Access to enhanced property predictions, routinely updated for all your compounds in your vaults. As we know, there's a lot of buzz around AI these days. What sets us apart is our focus on synthesizable molecules and comprehensive patent database search capabilities. This very pragmatic approach ensures that you're working with viable candidates and can quickly evaluate the novelty of your structures. Moreover, we offer API access to our deep learning technology, allowing you to integrate our AI capabilities into your own workflows and build your own predictive models. In essence, our AI module is designed to streamline your processes, enhance your insights, and accelerate your path to breakthrough discoveries. Now I'll hand it over to Roland for a live demonstration of those features. Roland, over to you for the AI demo.
Hello, my name is Roland Saito. I'm an application scientist with the CDD support team. Today, I'll discuss deep learning tools available to Vault users. The first feature I'll demonstrate is the deep learning similarity search. This is an internally developed application using a deep learning algorithm to search for molecules in Chembl, SureChembl, and the zinc library set. Chembl and SureChembl will find information like bioactivity and patent information. The zinc library will have synthesizable orderable molecules. The second feature I'll discuss is the deep learning bioisosteer tool. This is an application that generates synthesizable bioisosteers of your registered molecules in hopes that it can help with compound idea generation. So I'll go to my vault. I've registered a molecule. This is a clinical compound. It could be part of a commercial clinical compound library set. Perhaps you're doing drug repurposing. or it could be an internally synthesized molecule or part of a fragment or diversity set screen. You've come across some hit molecules and you'd like to know if those hit molecules or similar compounds have information associated with them. You can click on the deep learning link. You'll see a pop-up window of molecules that have been found in these various databases. Again, all of the searching is done within CDD Vault. None of your information will leave the system. Here I have Chembl and Sure Chembl toggled. You can also search the zinc library set. You'll see a zinc number. You can click on those links. It will take you to the enamine website, or you can put it into a cart and request a quote. Quite useful if you're doing a diversity set screen. You want to generate SAR around your hit molecules to see if it's an actual hit. You can use these zinc links to then accumulate molecules into the cart, and you can request a single quote. In your settings, you can increase the number of hits. You can choose to display the patent number. So if it's a Sure Chembl hit, there'll be a patent number associated with those hit molecules. You can choose to display the patent numbers that those molecules are in. You can also display the chemical properties and physical chemical properties listed here. You'll see them at the bottom. If the value is colored in red, the number is higher than your search molecule. If the number is colored in blue, the value is lower than your search molecule. So this is a quick way to use color to compare phys-chem properties with your search compound. On the bottom left, you'll also see a number. This is your similarity score. A score of 1 is an exact match. These molecules are sorted in descending order of these similarity scores. You can also choose to group the hits by scaffold. So the deep learning algorithm will recognize a core. So it's recognized this molecule and it's displaying the different analogs of this scaffold. So you have some fluorines coming off of this phenyl ring, a methyl off this piperazine. So we have a couple of ways to view your molecules. You can also launch the visualization session from here if you want to plot this information. You can also download the results. Now, if I click on the Chembl link, it will take me to the Chembl site where I can get more information about the molecule. There's a lot of information here. All this is within a few clicks of your search results. I won't go through them in detail, but here you can get information about molecule features, whether it's a first-in-class drug, if it's an oral drug. You can look at things like indication. The activity chart is quite useful. All of the compounds in Chembl have some sort of bioactivity data, so you can navigate your way to your activity charts. This might be part of a fragment screen. You might want to know if similar molecules hit different proteins. You can navigate to your activity chart. You can click on the chart itself. It will get you more granular information. If it's generating an EC50, you'll get the EC50 value, percent inhibition. You can get information about literature. So I can click on the chart here. I'll get more information about the document. You get the DOI, where you can then find your way to the original document if you have access to them. You also have a structural alert section. So in this case, it's recognized a sulfonamide. But if you're interested, if any of your hit molecules have similar compounds in the Chembl database, and if there's structural alerts associated with them, you can find that here. And lastly, you'll see a set of links at the bottom. If this compound is in other databases, here it's in the clinical trials database. It's also in Drug Bank, where you can get PK data as well as some pharmacology data. You might want to order this molecule from MedChem Express or get the zinc number. All of this information, again, is within a few clicks of your search results in Vault. Now, what the Sure Chembl site will get you is patent information. So this molecule is associated with multiple patents. If you click on the patent, it'll then take you to the actual document where you can download the PDF and get information within the patent. Now, lastly, I'll demo the BioIsosteer tool. If you click on the BioIsosteer link, it'll bring up a pop-up window. You'll see the molecule, your registered molecule on the left. Highlighted in blue is the area of the molecule that this tool is generating isosteers for. So in this case, you see a ring expansion with an oxygen inserted, things like ring contractions, different types of heterocycles that it's recognized. One of the take-home points here is that we are generating synthesizable molecules, and you don't have to sift through so many molecules and kind of weed out the bad-looking structures. We really aim to show only molecules that are synthesizable. This concludes the demo. If you find any of these features could help your research, please send CDD support an email, or you can contact your CDD account manager, and we'll be happy to activate the deep learning feature for you.
Hello, everyone. My name is Mark Garlinghouse, and I'm a business development representative for CDD. CDD has made important updates to the Vault inventory module designed to streamline laboratory inventory management. I will introduce some of those changes and then hand over to my colleague, Matthew, who will demonstrate this functionality in detail. The inventory module offers 3 primary features that significantly enhance efficiency. The first is advanced search functionality, a customizable alert system, and location-based sample association. The advanced search functionality enables rapid location of samples, reagents, or compounds. It also supports structure-based searches and queries across custom fields. And it integrates with other CDD Vault modules for comprehensive data access. The customizable alert system provides notifications for low stock levels and approaching expiration dates. It allows creation of custom alerts based on specific laboratory needs, and it facilitates proactive inventory management and regulatory compliance. The location-based sample association replaces traditional box-based systems with flexible location tracking. It allows the creation of custom nested locations, for example, freezer rooms, shelves, and racks. And it optimizes storage space utilization through occupancy tracking. Additional key features include a unique identifier assignment for each sample, custom fields for tracking specific sample attributes, and real-time updates accessible via a secure web-based interface. The inventory module is adaptable to various research domains, including chemistry and biology. For chemistry, it supports comprehensive compound documentation, chemical library organization, and hierarchical tracking from parent compounds to individual samples. For biology, it supports systematic logging of biological materials, detailed tracking for cell lines and tissues, and customizable metadata fields. We're excited about these enhancements and how they can help CDD Vault users be more effective in their jobs. Take a look for yourself at how the inventory module is designed to enhance research efficiency while maintaining data integrity and accessibility. Matthew, I'll let you take it away from here.
Hello, everyone. My name is Matthew Harris, and I'm a customer engagement scientist here with CDD Vault. For today's demonstration, I'll walk you through how a typical bench scientist might utilize CDD Vault's new sample inventory management to help with their day-to-day activities. Now, before we dive into the inventory management itself, we'll just briefly talk about how you can set up fields to start registering your samples in your Vault. The Vault administrators are the ones who will set up these fields for you, and that is done from the settings section under Vault and sample inventory fields. There are 2 areas here. You'll have sample fields, which will help you define the samples that you're registering. So maybe the tube type, any information that relates to the samples as they are being registered in the vault will go under sample fields, and the inventory fields will be able to track perhaps where it's located or any credits or debits that you'll be making to these samples. Now, a typical bench scientist may start their week looking for cells that they need to thaw for future experiments. How they can easily do that here within CDD Vault is by working from the sample search page here. Now, from this page, they can easily search for what they have on hand. Let's say that they're looking for their HeLa and A375 cell lines to thaw for their experiments. They can filter through their entries here, add a filter specifically on the cell type that they're looking for. As long as this is created as a sample field, you can easily filter on it here. And we can, from this list, select the A375 and HeLa cell type that we want to search for. We can now search in the vault, and we have 3 vials that match that description. So we have 2 HeLa cell lines and 1 A375, which are stored here in this liquid nitrogen location. It's very key to search before you go into liquid nitrogen because you don't want your cells to be out in the air for too long. Now, you can select, say, 2 vials that you need, so 1 from the HeLa and 1 from A375, and then you can take them from the liquid nitrogen. From the sample inventory here, these samples can be depleted, which allows you to remove them from the sample page and the sample kind of counted inventory. However, the record still remains within CDD Vault that these samples were created. So simply depleting the samples removes them from searching and also frees up that space in the box. So we can just deplete this HeLa cell line here. We'll yes to deplete it. And then also we'll deplete the A375 once we remove them for our experiments. Now, another task that a bench scientist may want to do is now search for compounds that they'll need to use in an experiment that they'll be running later in that day. It's very easy to do this as well. So if you have a list of, say, 3 compounds that you need to pull, again, you can filter through the entries that you have in your sample inventory based on the ID numbers that you have for those compounds. Here, we can search for those IDs, and I know I'll be pulling compounds 62, 63, and 64. Once I have those selected, I can search in my vault, and now they come up in my search. Now, the thing is, I'll need to pull these compounds as well as move them to my own compound box, which is separate from the box where they're located. So I need to perform 2 actions, taking a debit of the compound to run for my assay, as well as moving them. So that's very easily done from an Excel file that you can import into CDD Vault using the Import Data tab at the top. From here, I have an example where I've imported a file, taking again those batches and the samples specifically from the sample inventory. I'll be taking a debit of 25 microliters that I've mapped here in these 2 fields. And then for the locations, they'll be moved, instead of going to shelf A in that compound box, they're now going to shelf B in my own personal compound box. So these debit and change locations can be done very easily from the Import Data tab in one seamless step. Once that's completed, if I go back to now my sample search page and I look for my own personal compound box, I'll notice here that now I have those compounds moved, and I also have the debits that was taken from those samples. So the initial amount that was created was 50 microliters. I took 25 in that move that I just showed you, and now I have 25 left on hand. So it's very easy to move compounds within CDD Vault's inventory as well. Now lastly, let's say that you receive some compounds from a CRO that you now need to register in your inventory. If the compound box is full, it's very easy to create a new location from the sample inventory. We have that assay compound box, which is now full, but if we just select, say, the shelf that it's on, there's an option here for create a new box. This allows us now to add in a new box into the inventory locations list, and we can call this the assay compound box 2. And once we have that created, we can save it in our inventory locations and now update for others to now fill it with their received compounds. And that's all I wanted to show for the inventory module here in CDD Vault. You've been able to see how easy it is for a bench scientist in real time to interact with the inventory on hand and also create new spaces as compounds are shipped into their new locations. Again, if you would like a customized demonstration on how CDD Vault's inventory can meet your needs, or just more information, Please be sure to vote in the poll at the end of the webinar. And next, we'd like to introduce you to CDD Vault's Electronic Lab Notebook.
Hi everyone, I'm Tamsin Lamont. I'm a Senior Business Development Manager in the European team at CDD, and I'm excited to announce 2 new key updates to make our ELN even more collaborative and as efficient as ever. We now have customizable experiment templates so that you can create structured frameworks for any experiment type and seamlessly integrate text, tables, images, and files. This ensures consistency and reproducibility across your projects. We also have an advanced annotation tool. You can collaborate in real time with shared annotations, add critical observations directly within experiments, and you can customize workflows to fit your specific research needs. These new features complement our existing powerful capabilities. So we already have rapid data retrieval. Our robust search and filter functions make finding information really quick and easy. Intellectual property protection. We offer legal compliance features and audit tracking to safeguard your valuable research. And real-time collaboration. You can share findings and co-author documentation globally. We also have a nice Microsoft Office integration. You can access and edit Word, Excel, and PowerPoint files directly within the ELN without having to work outside of CDD Vault. Moreover, our ELN ensures regulatory compliance with features like audit trails and electronic signatures, which are crucial for meeting those GLP and FDA guidelines. Now, let's see all those features in sync in action. So Kelly, over to you for the demo.
Hi, everyone. My name is Kelly Bachochin, and I'm a customer engagement scientist here at Collaborative Drug Discovery. And today I'm going to be showing you the ELN module. So we are here logged into the Vault on this ELN tab. And this ELN tab is the index that shows you all the entries that have been created in your Vault that are associated to these projects on the left-hand side. If I were to uncheck one of these projects, then the entries associated with that project are then hidden from the list. On this page is also where I can do keyword searches as well as filter my entries by any of these metadata fields that you see here. Being a chemist, I have the ability to do a chemical structure or reaction search simply by clicking on this benzene icon. This will open up the structure editor where I can draw in a chemical structure. I can copy and paste from ChemDraw, or I can look up a molecule based on its common name, CAS number, or registration ID from my registration database. So here I paste in my structure. If I need to make a modification, I can update that structure. Then once I use this structure, the search will execute and find the entries where that substructure or reaction has been drawn. It will then highlight those in blue to show me the pages where that substructure is drawn. And I can then open up an entry simply by clicking on the link to open up an already created entry. If I did just want to simply create a brand new entry, I can use this create a new drop down and select ELN entry to start from a blank canvas where I can put chemical reactions or procedures for my biological assay or write down any notes in an ELN entry. But for now, I'm going to start from an already filled in entry. So I can click on this link to then open up that particular entry. Here I can add links to other pages in my ELN, or I can also add links to other parts of CDD Vault by clicking this insert link icon. Up at the top, I have a space for a title, as well as metadata fields that can be configured by my Vault administrator to capture information about the ELN entries themselves. So here I have a pick list to let me know that this is not a final product. I've set an experiment type to be a synthesis, and I can use these fields when I'm filtering back on that index page. Now here, since there is already a chemical reaction drawn, if I want to edit that reaction, I simply click into the reaction to pull up the structure editor and make any edits to my reaction that I need. If I wanted to insert a chemical reaction, I would use this benzene icon to open up the structure editor. Once I fill in a reaction, the system will recognize reagents that you've drawn next to, as well as above and below your reaction arrow, and it will generate a stoichiometry table automatically. If I want to then edit the amounts of each of my reagents, I click into the table to go into this edit mode, and I can change the amount simply by clicking on that amount and updating the amount that I've used and the units. Once I hit enter on my keyboard, the other amounts will auto adjust in the table as well. If I wanted to manually add a reagent or a reaction solvent, I can click either of these icons and use either a common name, a CAS number, or a registration ID as well. So here, if I type dimethylamine, it'll find it in my list. I hit enter and it adds it into the stoichiometry table. Now you'll notice any of my compounds that are drawn that are registered in my registration system, the IDs are automatically displayed, and I have the option to select a new batch or register a new batch or register a new molecule directly here from my ELN page. Underneath my table, I can click into the canvas of the editor and can start writing my procedure. And so if I just move over to an already filled in entry, you can see where you can start to add in headers and add links to your stoichiometry tables with that insert link icon. You can also attach files with this paperclip icon, or by dragging and dropping a file into the body of the entry. Now just moving over to a less chemistry focused entry, we do have previews of PDFs as well as images. So if you click on that file, it's going to open up the file in a preview where you would be able to zoom in and zoom out and view all of the pages, in this case, of this particular PDF once this loads up. Same thing for images. So if I click on an image, it's going to pull up a preview of that image where I can zoom in and zoom out of that image. But if I needed to download the image, I have that option as well. For files attached that are Microsoft Office files, you have the ability to expand the file and see the entire file here in line with your ELN entry. And we have an integration with those Office documents. So you can open those Office documents in your browser that you can make edits, and the edits will save in your entry once the page gets refreshed. And so if this were a file of data, then I wanted to import into my database, I have that option to do it directly from the entry. And doing things like importing data and registering molecules create links back to these pages once that data has been imported. Once I'm finished with my experiment, I have the ability to finalize or lock an entry, so edits can no longer be made. If you want, you can have witnessing turned on so that a witness needs to sign off on the finalization of this entry. And then all of the edits that are made to the page, as well as that witnessing, is going to be captured in the audit trail here, which is also downloadable. And that's the overview of our ELN. I'll hand it over to the next speaker. Thank you.
Thank you all for a great demonstration of our enhanced platform features. And a big thank you to all of our speakers today for their excellent presentations on our latest updates to curves, AI, inventory, and the ELN. And I'd also like to thank all of you in the audience for your attention and engagement throughout this webinar, because your interest and participation are what drive us to continuously improve our products. So before we move on to our Q&A session, I'd like to draw your attention to a poll that's now appearing on your screen. If any of these module add-ons we've discussed today look interesting to you, please take a moment to vote. You can indicate whether you'd like a customized demo or if you'd prefer us to send you some resources to learn more about these module add-ons. Your feedback here will help us tailor our follow-up to best meet your needs. Now let's transition to our Q&A panel. We've received some great questions in the Q&A box throughout the presentation, and we're excited to address them. So for those who haven't had a chance to ask questions yet, feel free to submit them now in the Q&A tab, and we'll do our best to answer as many as possible in the time we have remaining. So let's begin with our first question.
Hey guys, this is Eric Gifford. Thank you for joining us. We'll move into that question and answer session, as Lauren has pointed out. Again, my name is Eric Gifford. I'm joined with my CDD colleagues, Roland and Amelia, who are application scientists at CDD, and Dimitri, who is a business development scientist for CDD as well, covering ex-US, mostly Europe. Let's start in the way that the modules were presented. We had a few questions. By the way, this is a good news, bad news situation. Good news is we've got a ton of questions. Bad news is we don't have enough time, but we'll get answers back to everyone, and we can follow up with you individually as needed. Amelia, there were quite a few questions coming in about curves, and I wondered if you, because I do think your background is in biology. If not, I'm sorry for missing that. But I think you're the most appropriate person on the call here today to talk about curves and our implementation of those modules.
Yeah, let's see here. So we did get a few questions about curves, you're right. Really interesting ones as well. Should we take the first one?
Yeah, it's fine. I mean, just as an overview, it's an amazing the number and the variety that we're able to handle in CDD Vault. And I wondered if, some of these, what used to be novel or unique or, outlier types of assay formats, they're now becoming mainstream, and we can actually accommodate that type of data analysis now. So what are your thoughts on curves? Maybe in general and more specific questions if they come to mind.
Right. So for curves, I'd say the functionality that we have released recently, it adds a lot to the flexibility of the platform. So it's not just your typical sigmoidal curve that you can plot, and not just, using raw values. You can actually use calculated values in your plots on the actual axis as well. Just a lot more options that you can actually use to plot a lot more of a variety of data, which is really, really nice. And also, since the parser was mentioned, what's really important is that you can actually take your files sort of directly from your instrument, put it into CDD Vault like we saw demonstrated already today, and you can just plug it in there, let CDD map it for you with the template, and you can just put your data in really easily, get it plotted, and get your calculations out really quickly.
That sounds good. And as application scientists, I know you're tightly connected to our development team. And I'm wondering what your thoughts are in terms of, we see two. We had one before, right? It was the hill slope. Now we talk about the bell curve. And do you see this expanding? Do you see an expanding universe of different curve types? And I think the other interesting fact is that it's all embedded right within CDD Vault. You don't need to go outside to another tool to do this, right? It's just right there in your Vault.
Yeah, so we do have more curve fits coming out in, I'd say, the near future as well. So don't think that, our recent bell curve development is the end of it. It's definitely not. More is coming out. So stay tuned for that, definitely.
And Dimitri, I guess that's a good segue to sort of the, how do features, be it in curves or other components or features of CDD, how do they get entered into the product roadmap? I think you have some words and maybe experience in taking customer feedback to our developers.
Thank you, Eric, for passing the ball, and thank you everyone for attending. I want to say before jumping into the feedback section and the updates about the curves that I still remember expressions from customers, seeing the overlay of different molecules and their happy smiles. And we'll make sure to continue that, offering more features. Regarding the feedback, it's essential, it's significant. Receiving the feedback from customers, this is exactly what drives the CDD Vault roadmap. Every week, we meet internally, discuss what we received from our more than 600 customers across the globe. And with the product team, we count votes pretty much of what's coming next. So please to the community and customers, we are always delighted to listen to the feedback from customers to evolve together CDD Vault. Eric, so passing the ball about the feedback, of course, this is done with the sales team and the support team. Any more thoughts from you, Eric? There might be a connection issue with Eric. Roland, Emilia, all good from your side?
Yes, I think
Yep.
Eric has just unfortunately frozen for hopefully not too long, but yeah.
I'm sure she wants to say two things. Eric, are you back?
I think that we've got a quarter for the weak, perhaps weak connection here. Let's move on to some of the other components, and thank you, Dimitri, for that. We've got AI. Roland, thank you. You did a fantastic overview of that. I want to point out to folks that we have what we call the scientific CDD webinar coming up just in a couple weeks. So September 26th, if you want to do a deep dive into AI and how CDD is evolving the science around the AI modeling, I encourage you to register for that webinar. We can take a few questions about that, but I really wanted to move over to inventory. And Roland, talk to you about some, or maybe you've been monitoring the questions, and you could address some of the questions that we have for inventory and what you think the high points are, the key features for the new inventory module.
Yeah, so I'll be happy to take inventory questions. So we got a question about being able to track solids or liquids for the same batch. And with the new release of our inventory module, you can create multiple samples per batch. You can create custom fields. You can create pick lists to indicate whether they're solids, neats, liquids, really any descriptors for your samples, including barcodes. You can use your barcode as your sample identifier. You can indicate whether it's a solution, and if it is, concentrations, molarities, solvents it's dissolved in. All of this information is going to be within your hands. One of the recent releases was the inventory search. So if that's not activated in your Vault currently, please contact support or your CDD account manager, and we can activate inventory searching for you. Let's see, we've also had a question about having a inventory storage unit and having multiple units that might have similar directory or a tree. You can absolutely duplicate your inventory trees, including boxes that you may have within those inventories. The Vault admin can create these directories. One of the options you'll see when creating a location is to be able to duplicate the entire location tree. So if you would like support on how to do this, again, please contact support and we'll be happy to walk you through it. But you can also find that within your settings. It's under Vault Sample Inventory, and in the locations, you can edit your locations and duplicate your location tree.
It's a really interesting feature, Roland, and something that I see that goes across CDD Vault. And it's listening to feedback. It's this notion that you can share things, duplicate templates, right? So, even from curves when you're uploading data, we now have the ability to very easily share what I'll call templates as a non-technical person. But just like you've said there, you can replicate location trees and push that across. I think that's also visible in our ELN, right? So it's a bit of a segue into the ELN and how we really rely on ELN as the place to enter data, share data, and mine data then by bouncing out to something like visualization or curve fits. So Amelia or Roland, what gets you excited about the current or the most recent versions of ELN, and maybe a bit of a teaser? What do you see coming up that is like gonna really knock people's socks off?
Maybe if I can start here, just kind of a few things that I want to point out. You did see them during the demo, of course, but what makes me the most happy about the new ELN is the previews for the images specifically, because since I am more on the biology side, I usually record or do examples of assays recorded in the ELN, and I do have a lot of images that are there, sometimes with very small text in them that I couldn't really read before, even with 3 sizes. So now that you can actually preview them, it's quite nice to just look at them directly in the ELN. You don't have to download anything out or put it back in. It's just quite nice to do it all on the one page, and same for the PDFs as well, to actually maybe attach an article and read it in there. It's quite convenient.
And Roland, from a chemist perspective, yeah.
Mhm. So yeah, I do like those features as well. The image previews and the PDF previews, being able to dig in through the document without having to actually download the document to your downloads folder. I think that's a great new feature. Being able to merge cells, indent bullet lists and numbered lists. These are all new features. On the chemistry side, we also have additional features. We don't have time to go through all of them, but one of them is the way we handle precision with numbers in the ELN now. Masses, the way we've increased the number of significant digits when calculating your molecular weights, handling with more precision the number that you enter for masses or in your stoichiometry table. There's a whole host of new features that are released in the pre-release, which is going to be a full release soon. But it won't stop there. This is going to be an ongoing development for CDD, is to continually improve the ELN.
Yeah, and I see Andreas just popped a question in there, which I was thinking as well. So, hey, must be a good question, right? Joking. But do we need to activate? How do you activate the new ELN? Same as always, call support, account manager, or is it just on, right? Tell us a little bit about the process for turning on any of these add-ons, these new modules.
Yep, you can either contact CDD support or contact your account manager, and we'll just flip a switch and turn it on for you. It's quite simple.
But what do you think? So Dimitri, you're an account manager. What's the conversation that you usually have? I'm Eric, the customer, and I say, hey, Dimitri, I really want to try this. This thing looks great. What would your response to me be?
These points are always exciting for me, receiving them from customers, right? Turning on features, it might be from the inventory for the new inventory, the ELN, the AI, all those needs to be individually turned on. The ways, of course, are either the account manager or the support at collaborativedrug.com, reach out to support. It's always exciting, turning them on and then scheduling, for example, a meeting with your customer, discussing, going through them with a support team, the CDD support team, and engaging with the customer, receiving feedback, receiving what we can do more to develop the feature. So excitement, engagement with customers, with our community, I think makes us mucho happy, right? We help, they get the support to progress, and puts a smile on our faces. Simple as that.
Yeah, well put. And a shout out and a big thank you to our support team. And I hope customers realize that the support team is always on call. They're just a phone call away, just an email away at no cost, right? Which is such a differentiator for any other solution out there. I'll say that directly. And don't hesitate to reach out to support or your account manager. We're here to help activate any of these new modules and provide training, even if you're not subscribed to them. But you just want to learn more about it. We can arrange a training session with you so that you can learn more about that. Anything else? Questions that have gone unanswered? Pop them in the window. People have anything else to say on this? We could go back to the AI, but I really want to save that for our 26th webinar, but I'll just pop one out there. So Roland, you mentioned the ability to identify scaffolds and to look at the SAR and that type of thing. What's your thinking as a chemist? I mean, how important is a tool to suggest ideas around compounds that aren't, let's just say, crazy ideas, right? They're actually rooted in some sort of reality, meaning you could probably go buy that compound or something very, very similar to it. How important is that to you as a chemist?
Yeah, being able to look at a set of molecules and not have to sift through things that can't be synthesized or just look just funny to a chemist. We really try to present molecules that seem interesting and that are synthesizable. And hope so, this is not going to replace your chemist. It's really so your chemist can look at the molecules and say, oh, I didn't think of that, or really just generating ideas faster. As a chemist, for me, it's easy for me to just sort of walk nitrogens around the ring or stake fluorines, switch a hydrogen to a fluorine. But sort of the bigger changes, changing the heterocycle, changing how things are connected, we really try to present you with a wide variety of isosteres to hopefully speed up the drug development process.
That sounds good. And once you have those, yes, go ahead.
Something to add here, receiving it again from customers, right? The feedback is doing all this from CDD Vault, from the secure environment of CDD Vault platform, and fast. These two parameters it might be so logical for us, right? But we can make it happen securely, first of all, and fast. Key for success.
Very, very true. And I want to, go off that point into a question here. It's about, so once you have these ideas for compounds, you can actually buy them in some instances. But, a lot of times, if you're in a small startup, even big companies like the ones I worked at, you'd go to CROs. And there is a question here is, can we use the ELN to collaborate with a chemistry or biology CRO? Right? And I think that drives right to the heart of collaborative drug discovery. So maybe, well, anyone, what's your thinking on how this package, this platform enables collaborations amongst scientists, both internally and externally?
I can take it because the bigger the community, the stronger the collaboration is inside the Vault. First of all, it's achievable through projects. You can easily share a specific data set collaborate with a CRO. It might be expanding it, Eric, with investors, with consultants, anything, any direction you like to take. So you just give a specific user access to your collaborator, invite them in a specific project, sharing a specific data set, and then you can collaborate only there, right? Keeping it siloed the rest of your data set. And achieving at the end live collaboration. This is what we're hearing, achieving live collaboration instead of just sharing spreadsheets via email, via share drop, inside a specific drug discovery data management platform. Makes it easier and secure.
Yeah. So one of the things, and that secure is a key word, right? It's right in the name of the product as well, Vault, right? So secure. And I think it's worth mentioning the flexibility that users have, or Vault administrators specifically have, to turn on and off features at the individual level through projects. And maybe Roland or Amelia, you want to talk about, something as simple. It's not simple to implement. It's simple to say, but it's hard to implement and do, is what I'll call structure masking, right? There may be, pieces of information that I don't want to share with CROs, right? Maybe I don't want screeners to see structures, right? Maybe I don't want chemists to see biology data, right? Talk a little bit about that granularity of being able to view the data depending on your role.
Yeah, so I can touch a bit on that. So yeah, definitely if you're working with a CRO, it's quite common that if they are providing assay data results for you, they might not need to, or you might not want them to actually see the structures. And that can be achieved with projects, but previously these structure masked users, or if you want to do the structure masking, you can of course hide the structures, or you can try and achieve the hiding of assay data through projects. So there's two ways you can do that. But for structure masking specifically, you couldn't access the ELN before, so that was just completely unavailable for structure mask users. But now you can actually use it, which is quite nice. So you can still kind of share notes, share the process of how you do things, and just still hide the necessary information there, which would be the structures.
Roland, anything to add there? We got about 1 minute left, so this might even be the final
That was
few
a
words.
great answer.
I think that was a huge leap forward in terms of our ELN. And again, it responded to multiple customer requests to put that structure masking into the program. So thanks again to everyone. We're at the top of the hour. We had fun. It went really fast. That's how I know it was it never long enough. We've got some more questions. And you're happy to please take the poll. Once you get your name on that poll, we'll follow up with a demo or just more information. And you can always reach out to us through info@collaborativedrug.com or support@collaborativedrug.com, and someone will be back in touch with you. Thank you all very much.
Thank you.
Thank you.
Bye-bye.
Thank you all.